Hello students, today we will be studying the cell division. Cell division is the process by which a parent cell divides into two or more daughter cells. This usually occurs as a part of a larger cell cycle. In eukaryotes, there are two distinct types of cell divisions, mitosis and meiosis. Now, mitosis is a vegetative division wherein each daughter cell is genetically identical to the parent cell, whereas meiosis is a reductive cell division. The number of chromosomes in the daughter cells is reduced by half to produce haploid gametes. Now, let us first see mitosis. Mitosis is the process by which a cell which has previously replicated each of its chromosomes, separates the chromosomes in its cell nucleus into two identical sets of chromosomes, each set in its own new nucleus. It is a form of nuclear division. It is generally followed immediately by cytokinesis, which divides the nuclei, cytoplasm, organelles and cell membrane into two cells containing roughly equal shares of these cellular components. This type of division occurs in all somatic cells and in immature germ cells and is the most common method of cell division. The entire process takes about 1 to 1.5 hours to complete. The daughter cells have the same number of chromosomes and genetic distribution as is seen in the mother cell. Hence, mitosis is called as homotypical or equating division. Before the cell divides, there exists a period known as interphase, during which the individual chromosomes are not visible, as they are uncoiled into very long and narrow chromatin threads. The nucleus of the cell during interphase possesses a nuclear membrane containing the network of chromatin threads or granules and a nucleolus. Replication of DNA takes place in the chromosomes. A pair of centrioles is present just outside the nuclear membrane within the cytoplasm. Now, this here is a picture which shows us the various stages of mitosis. What we see here is the interphase, where we can see that the chromosomes are seen as thin strands, nuclear membrane is present. Then next we go on to see the 4 successive stages of mitosis, and these are prophase, metaphase, anaphase and telophase. Prophase. Herein, individual chromosomes can be visualized under the microscope as long, thin threads as they are tightly coiled along the entire length. Each chromosome splits longitudinally into 2 chromatids except at the region of the centromere. The 2 paired centrioles are separated from each other by elongation of continuous microtubules, and this is known as the achromatic spindle, and this occupies the opposite poles of the nucleus. So what we see here is the prophase stage, where we can identify the chromosomes as having a pair of chromatids connected by the centromere. The continuous microtubules are organized by the centrioles. Later, the nuclear membrane and nucleolus disappear. The chromosomes now get entangled in the meshwork of continuous microtubules, and this stage may be called prometaphase. What we see here is the prometaphase stage. The nuclear membrane and nucleolus have disappeared, and the microtubules are attaching or They are responsible for these chromosomes to lie in the equatorial plane and also to attach these chromosomes to the centromeres. This is the pro-metaphase stage. Next, we see the metaphase. At this stage, the centromere region of each chromosome presents a bilateral disc-like kinetochores. From these kinetochores, a set of chromosomal microtubules get organized and extend towards the centrioles. Growth of the chromosomal microtubules allows the centromeres of the individual chromosomes with their paired chromatids to occupy the equatorial plane of the spindle. This gives rise to the ester formation in the metaphase, and this stage lasts for about 20 minutes. What we see here is the metaphase, where we see all the chromosomes, they have aligned in the equatorial plane and the microtubules are attaching it to the centrioles. The next stage is the anaphase stage. In this stage, the centromeres split longitudinally and the two chromatids separate to form two new chromosomes. Each pair of the newly formed chromosomes separates and migrates to the opposite pole due to the contraction of the continuous microtubules of the spindle. This is what is seen here in this diagram, where the centromeres have split so that the individual chromosomes are separated and they are pulled towards the two poles, or they are pulled towards the centrioles. Next comes the telophase. Herein, the daughter chromosomes are now enveloped by the nuclear membrane and the nucleolus reappears. The chromosomes get uncoiled and are difficult to identify under the light microscope. This is followed by division of the cytoplasm and the two daughter cells are formed. So what we see here is the telophase stage, wherein the nuclear membrane will now reappear, the nucleolus reappears, and then there will be the division of the cytoplasm and the cell organelles, and finally that is going to give rise to the two daughter cells. So this last stage which is seen here is the cytokinesis, that is division of the cytoplasm and other cell organelles and formation of the two daughter cells, each having 46 chromosomes. So daughter cells are similar to the parent cell or the mother cell. Another picture showing us the different stages of mitosis. So what we see here is the prophase stage, the metaphase, the anaphase and the telophase. And when the telophase gets completed, we see the two newly formed daughter cells. Now let us see what are the features of mitosis. So mitosis occurs in all somatic cells and in the immature germ cells. Daughter cells are completely identical to the parent cell. Each division is preceded by interphase, during which the DNA replicates. Now we go on to study the meiotic division or meiosis. This type of division occurs only in the sex cells during the maturation of the gametes. Herein, the diploid parent cell gives rise to haploid daughter cells, and this is also known as heterotypical division, and it consists of two consecutive divisions. They are known as the first meiotic division and the second meiotic division. So let us now see the first meiotic division. This has four successive stages: prophase, metaphase 1, anaphase 1 and telophase 1. Prophase. This is a prolonged stage and further can be divided into 5 substages. Leptotheme. Herein, the individual chromosomes are seen as long, thin threads having beaded chromomeres along their length. Next is the zygotine stage. The homologous chromosomes are arranged in pairs lengthwise with a point-to-point alignment. This process is called synapsis. Each chromosome pair has one maternal and one paternal chromosome. Next, we see the Pachytene stage. Each chromosome splits longitudinally into two chromatids except at the centromere region. Thus, 4 chromatids are arranged in a row, giving rise to the tetrad formation. The chromosomes progressively get shortened and thickened. What we see here in this picture is the Pachytene stage, the Diploidine stage. Next, we come on to the Diploidine stage. In this stage, the non-sister chromatids of a tetrad cross each other at one or more points and form the chiasma. At the point of crossing over, the chromatids break with the help of endonuclease and then unite with the segment of the non-sister chromatid with the help of enzyme ligase. And this process is known as the crossover. And it is this crossing over which permits continuous reshuffling of genes and the formation of the structural basis of hereditary diversity between the offsprings. So what we see here is the tetrad formation, and then we see that the non-sister chromosomes have crossed and then they get connected. They detach and then they unite, thus giving rise to reshuffling of the genetic material. Next, we see the diakinesis, which is the last stage of prophase. In this stage, the homologous centromeres pull apart and the chromosomes are separated after uncoiling. The nuclear membrane and nucleolus disappear. Next, we go on to see the metaphase 1. After crossing over, the homologous pairs of chromosomes align in the equatorial plane of the spindle. Next comes the anaphase 1. In this stage, the centromeres do not divide. The homologous pairs of chromosomes separate from each other and migrate to the opposite poles of the spindle, carrying their pairs of chromatids. So as is seen here, the entire chromosome has gone to the opposite poles. There is no division of the centromeres here. Crossing over has already occurred, so reshuffling of genes has already taken place. The entire chromosome will move towards the opposite pole. Next, we go on to the telophase 1. Herein, the cytoplasm divides and the two daughter cells are formed. Each of the two daughter cells that are formed contain half the number of the original chromosomes. That is, they are haploid. The first meiotic division is therefore referred to as a reduction division. So that's what's seen here in the telophase 1 stage, where the entire chromosome along with the nuclear membrane is restored and it has got haploid number of chromosomes. Next, we see the second meiotic division. Each of the daughter cell of the first meiotic division enters directly into metaphase 2 and the chromosomes align in the new equatorial plane of the spindle. Anaphase 2, the centromeres divide and the chromatids separate to form new chromosomes which migrate to the opposite poles of the spindle. So what we see here is the metaphase 2. Herein we see that there is splitting of the centromere, and then in anaphase 2 we see the chromatids moving towards the opposite poles in both the daughter cells of the first meiotic division. Finally, the telophase 2. This gives rise to 4 haploid gametes from the 2 cells of the first meiotic division. The polynucleotide strand of DNA molecule having the typical double helix structure is formed. The second meiotic division is similar to a meiotic division. That's what we see here in telophase 2, where we have the 4 daughter cells which are formed and each of them is haploid in number. So the features of meiosis would be appearance of chromosomes and pairing of homologous chromosomes occurs in early prophase of the first meiotic division. Crossing over during late prophase permits a continuous reshuffling of genes. In anaphase 1, centromeres do not divide, thus giving rise to reduction division of the first meiotic division with haploid number of chromosomes. In anaphase 2, centromeres divide and this maintains the haploid number of chromosomes. 4 haploid daughter cells are then transformed into functional gametes, that is either spermatozoa or ova. Reduction division in meiosis is a must. Only then an ovum, when fertilized with a spermatozoon, will give rise to a zygote having diploid number of chromosomes, which is constant for the particular species. Thus, we have studied the two different types of cell divisions today, mitosis and meiosis. Thank you.